All right, you can see that okay?
Perfect.
Thank you. So thank you, Jorn, for that wonderful introduction. And indeed, today I'm going to provide an overview of the work that's been ongoing for over a decade to generate reference epigenome maps to drive research, both in discovery research and translational research in human epigenetics. So in terms of the overview of what I'll be talking about today, I'll start by introducing IHEC, so the International Human Epigenome Consortium, and the EpiATLAS resource. And then I'm going to really touch on two brief vignettes. These are unpublished stories from two graduate students in the lab, which would give you some indication of some of the applications of the EpiATLAS. So first, I'll talk about probabilistic epigenomic references as a unifying framework to define epigenetic variation, both in normal development and disease. And here you can think about this as a way of really generating a framework in the same way that we have the human reference genome. If we can think about the human epigenome at a cell type specific level to generate references that allow us to generate a framework to then describe variation. And then I'm really briefly going to talk on another topic, which is defining cell type specific interpretation of common and rare variants. So EWAS studies and others typically look at heterogeneous populations, but the EpiATLAS allows us to go into single populations that have been self-sorted to 94-95% purity and ask similar questions, but ask whether cell type interpretation of variance is a common mechanism in human, in this particular case, human breast. So what is IHEC? So IHEC is an international consortium. Involves three main executive groups: the executive committee, which is composed of funding agency representatives. We have the which communicates with the International Steering Committee, and through these two committees, we work in focus groups, which we call working groups. And the objectives of the IHEC consortium is really to coordinate the production of reference maps for human genomes. And again, the founding member of IHEC was the Roadmap Consortium, and many of you probably, hopefully, are aware of the 2015 marker paper from Roadmap, followed up by the 2016 publication package from Blueprint, and currently we're working on an update to that. And really, our role is to coordinate the rapid distribution of data to the entire research community, and I'll talk about the process in which we're doing that. And also to try and coordinate a minimal amount of redundancy across different epigenetic efforts around the world. So really, we're building on a legacy. So what do we define as a reference epigenome? Well, a reference epigenome is a description of a genomic state. So obviously in our cell, in our body, we have over 300 distinct cell types, and each one of those cell types, as far as we're aware, has a distinct epigenetic state. So like the reference genome, which we have one reference genome for the human species, although, as we just saw announced yesterday, I think it was, the pan reference genome really bringing us forward into integrating genetic variation in the human reference genome. In epigenetic space, we really need to generate these references at a cell type specific level. And of course, that's just the beginning. That just provides a framework to begin to understand the variation within humans, and then of course temporally across time, and then of course as human diseases emerge, as disease emerges. So the EpiATLAS itself is composed of deep RNA sequencing. So this is 200 million reads, typically ribodepleted RNA sequencing, whole genome bisulfite sequencing. This is typically done on an Illumina platform. And then there's a core set of ChIP-seq data sets generated, and those are listed there. And that set of 6 marks was chosen based on redundancy across the 100 or so histone modifications that we are currently aware of. And so these are the set that really describes the space in the most comprehensive way given the limitations, obviously, in sequencing. So there are 10 consortia that have contributed to the International Human Epigenome Consortium shown here. The workflow is essentially generating these data. We then deposit the data. This is all human subject data, so it's for the most part protected and needs to be put into protected repositories, including DDBJ, the EGA, and dbGaP. Although there are some rare unprotected data sets through the ENCODE consortium, which are included in the reference. These, once deposited, are then downloaded, and I'll talk about that, and staged, where we download the metadata, the FASTQs, and import into a local database. We then run containers, and so these containers are designed to allow for distributed comparative analysis of your data set against the reference, and then we post that data through various portals, including the IHEC data portal. So as part of the IHEC reference, as part of the IHEC consortium efforts, we've really been focused on generating standardized experimental and analytical workflows. So just as an example, here is a standardized experimental workflow in place now at the University of British Columbia and BC Cancer, where we've designed a workflow that allows us to do reference epigenome mapping, in this particular case, on resected tumor specimens. And here, this is both in the context of needle core biopsies, but also in bulk resected tissues. And you can see the various steps that we do. First, central path review. Secondly, liquid nitrogen pulverization. We then do essentially a dual RNA DNA extraction to allow us to quantitate the amount of chromatin present in that specimen. And from that, we then go into a native ChIP-seq protocol that we tune based on the density of nuclei within that particular specimen that we're working at. We've also generated standardized container pipelines, and these provide opportunities for distributed comparative analysis. So you can imagine, with this data behind protected repositories, in fact, the vast majority of human epigenetic data indeed exists in this manner. It is actually, as I will show you, quite complex and time consuming to download this data and reprocess it yourself. And so by providing these IHEC containers, it allows individual users across the world to run their own data through the container and then compare it directly to the references, and therefore bypassing any analytical differences between the two, between your data or the user's data and the IHEC references. And as part of this, we have designed or we have implemented what we call the epigenomic reference registry. Now, it may come as some surprise to you, but there's actually currently within the protected repository framework, including EGA and the EGA nodes, there is no way to search across the repository for protected human data sets, epigenomic or otherwise. So if you wanted, for example, to ask the question, well, how many H3K4 trimethylation data sets exist within the protected repository? This information is not available because you must first ask for DAC permission to be able to view the metadata that's behind the firewall. And so what we have designed is a process by which de-identified metadata in a standardized format can be made available for public searching, which then allows you to essentially traverse across these data sets and then select the data sets that you might want to continue processing. In this particular case, these data sets that are currently part of the EpiR registry are actually all IHEC data sets. But indeed, the plan as we move forward into 2023, 2024 is to make this portal available to epigenetic researchers across the world and allow others to submit to this repository. And again, we have generated a standardized metadata framework for this and a validator to facilitate community submissions. So just to sort of touch on and maybe linger on this slide a little bit, you know, I think this really highlights a significant gap in our current framework for protected data sharing. So this just gives you an actual timeline of how long it has taken our team to download and reprocess the IHEC reference data sets, keeping in mind that we're part of the consortium. But the process that we undertook here is going through the EGA in this particular case, and or dbGaP and ddJB, and going through the DAC permission process. I could spend a lot of time talking about some of the challenges there, but one of the main challenges we faced is actually doing a large-scale data transfer or data download from the EGA itself. It is really not designed for large-scale data transfers, and we had to generate customized scripts that would essentially continue to query the database because we would have pipe failures, and we would have essentially drops of the connection, and we'd have to reconnect over and over and over again. And in fact, we had a process where we would reconnect up to 10 times to allow the downloads. And even that, it took literally months in some cases to download these data sets. So this is an issue, I think, for our community that we need to continue to work towards designing a sharing framework for human protected data that's going to enable research into the future. So what is it that we have generated? This is the sort of high-level view of it. There are just under 3,000 human subject data sets. So these are individual either cells or tissues from human subjects that have been subjected to the set of assays that are shown here. The dark blue indicates observed data. The lighter blue indicates imputed data. And so we have now imputed across this entire data set. And this is part of what we're calling the EpiATLAS. You can see, not surprisingly, we are enriched in blood cell types because this is a human tissue that's readily accessible and readily pure and can be purified using standard FACS sorting modalities. And of course, this is the tissue type that was a focus of the DEEP consortium, or sorry, of the Blueprint consortium. The EpiATLAS itself, when we traverse across these data sets, and here I'm just showing you some examples. This is H3K4 trimethylation, where we've asked, does the promoter occupancy of H3K4 tri replicate biological relationships? And indeed it does. And you can see the bifurcation as well annotated in the literature between blood and this blue. So blood is a very distinct epigenetically across all marks, in fact, compared to the solid tissues that are shown here. And on the right-hand side, I'm showing you an example just to focus in on, these are purified cell types from the breast. So these are the three mammary epithelial cell types. And then in this pink here is in the blue, sorry, is the stromal cells. So these surround the mammary duct structure. And really the point here is that indeed we can recapitulate. This is across 8 individuals where these cell types have been purified as part of the reference. And then I just wanted to point out here in this sort of, I don't know what color this is, greeny yellow color, that cell lines, and these are well-studied breast cell lines like MCF10A, that these have a very distinct epigenetic state. And so I think we have to reflect on the fact that if we're studying epigenetics, that really we need to be studying it in the context of the primary tissues and cells, and be very cautious in interpreting any epigenetic states that we observe within cell lines. As you can see here, very distinct. So in terms of the breakdown, there are assays depending on the particular interest of the consortium members. There are assays there are cell types where we have individual assays. For example, we have just under 300 brain samples where we only have K27 acetylation available. This was to do a quantitative analysis of enhancer regulation in this particular assay. But you can see that we also have a good distribution of cell types where we have all 8 core assays. And again, there are some blood cell types as well, where we have individual assays or in some cases combinations. As part of the IHEC reference, we've also generated standardized metrics. And I think this is something that will be very helpful, or I hope will be very helpful to the research community. We call these quality metric summaries. We generate 5 per assay, and we compute these across all of the IHEC references. And so if you are doing an epigenetic assay in your own lab, or you've downloaded one from your protected repository and you want to compare its quality, you can compute these metrics, or they're computed for you within the container, and then compare them against the IHEC reference. And so these are just very simple. These ones are just depth metrics. We have minimum standards in terms of depth. So 25 million fragments, so that's 25 million single-end reads, or 15 million single-end reads for punctate marks, 50 million or 100 million paired-end reads for K27 trimethylation. So this is another view of a quality metric summary, in this particular case, something called FRIP, which is where we compute the fraction of reads that align within a FDR 0.05 cutoff max peak. And we can see that we have a distribution of qualities, and some of these distributions we can see cluster by or have indication of a bias within the different consortium. You can see, for example, here, this blue color versus this green color, I think is a good example. And really, I wanted to show this is just to highlight the fact that within the references, and indeed this is true across all epigenetic assays that I'm aware of, we get a distribution of qualities. Some of these at the very end of the distribution are very hard to reach tissues, unstable tissues, and these affect the quality of the assay. And indeed, in the reference, we have used these data to then qualify samples as either essentially pass, or we're not using the word fail, but level 1, level 2, gold, silver, that type of thing. Okay, so just that gives you an idea of what's in the reference. We're looking forward to pushing the reference out. We're actively analyzing it now, and we're hoping to have this out for the community by the fall of 2023. But I'm going to give you just a few examples of some of the applications now in the last 10 minutes or so of the types of things that one can do. So one of the challenges with this distribution in FRIP score, so again, this is looking at the distribution in fraction of reads and peaks. Obviously, the higher the number of reads that are recruited to peaks, the better quality the data set is. In other words, the signal to noise is much higher when we see more of the fraction of reads being recruited to peaks versus the background. And so this distribution creates an issue because we have a variety of false negative and false positive calls within a given cell type. And so that's kind of listed here. And so really what Jonathan set out to do is to develop a computational model to estimate the probability of histone modifications occurring. So rather than a binary call, when we have multiple observations for a given cell type, this gives us an ability to actually generate a probabilistic output for a particular bin in the genome that would indicate within that cell type whether that particular feature is present or not. And I should say that Jason Ernst's group has also, I think, recently published on bioRxiv a similar approach using, I believe, using the roadmap data to begin with. So you might be interested in looking at that. So the method that Jonathan developed is basically a Bayesian hierarchical model. This just gives you an example of kind of a visual example of the challenge. So you can see that we have observed peaks down at the bottom. And of course, the observed peaks that we see are a combination of both false positives, well, true positives, but also false positives and false negatives. And really what the Bayesian estimate is trying to do is to identify both the false negatives and true positives, and then generate a theta that would represent a probability that in that particular bin, the particular modification is present. And so when we run this on simulated data, we can see that it performs as we might expect. That the root mean square error of the model estimates decreases with the number of genomic positions that it's trained upon, and we see this sort of plateauing or stability as we increase the number of positions. And when we compare it here in the bottom panel, to the true theta, we can see that the Bayesian estimate does a slightly better job, but certainly a significantly better job in many cases versus a maximum like a naive maximum likely estimate. So we think the Bayesian estimate is doing a better job of predicting the false negative and false positive occupancies for these genomes. And so we can take this model and then fit it across the EpiATLAS ChIP-seq data to identify data sets of low quality, or to see how it performs with respect to, in this particular case, low quality as defined by, again, the fraction of reads within peaks. And you can see indeed that the estimates improve as the fraction of reads and peaks increase, as might be expected. And this allows us then to identify data sets that are circled here in red that cluster inappropriate in a UMAP and also perform poorly in this assay. Okay, so that's sort of one aspect, and that probabilistic reference may provide, or arguably could provide, a unified framework to then compare any given data set against, because it's going to allow you then to, if you do a ChIP-seq data set, or you have a reference that you've generated for your own cell type, it would allow you then to compare that to the reference and annotate any variation that you see versus this probabilistic reference. In the same way that over the last 20 years, we have generated SNPs, or single nucleotide polymorphisms, or more recently, large structural changes with respect to the reference. That ability to compare to a reference has really driven our ability and our understanding of genetic variation in the human genome. And I think in the same way that the epigenetic community really needs to move forward to the concept of having a reference to allow us then to compare against, and then to allow us to be interoperable across the community. So the second story I'm just really going to briefly touch on here is the impact of cell type on genetic and epigenetic interactions. So I think conceptually this is fairly easy to understand. We understand that there's epigenetic states that define cell type differentiation and development. But in addition, we have genetic variants that are being, or arguably, or the hypothesis is that there are genetic variants that exist within a genome, and these are germline, that are being interpreted in a cell type specific manner. In other words, a transcription factor that is recruited to a particular element is only present in one cell type and not another. And so that variant that may be within the binding element of that transcription factor would therefore influence its ability to its affinity to the DNA, and therefore influence the ultimate outcome. And so this is kind of a simplistic thing on the right-hand side, showing that indeed in cell type A, you see no response over this heterozygote position, but in cell type B, you see a difference. And so the system that we're using to ask this question, of course, this question has been asked to some degree in blood cell types, but as far as we're aware, has not been asked across solid tissues. And so we're leveraging the mammary system here because it allows us to purify cell types from normal women that have been generated, that have been obtained through reduction mammoplasties, and we can sort for luminal progenitors, luminal cells, and basal cells, and then the stromal cell component of the human breast. And this just shows you the individuals, and in fact recapitulate the relationships as we expect, including the relationship between the luminal cell, the luminal progenitor cells, and the mature luminal cells. And then the basal cells that are shown here. So this is the framework we're using. And for these women, we also did whole genome shotgunning of the germline. And so we have variants, and then we can use those variants to essentially do a reference bias robust alignment with the ChIP-seq reads. So we corrected the reference for the variants that are present in these women. And then we did what we're calling cis-hBTL, so binary histone trait loci. And from there, we associated those to genes and then looked for examples where we had a change in the histone occupancy associated with a heterozygote variant that was also associated to a change in expression in a nearby gene. So this is just a representation of all the variants. So there was something like 7.5 million variants across, that was called across, this is the union, of course, called across all 8 women. And when we looked at the individual variants across the cell types, you can see that the majority of these variants were associated with unique cell types. So here, 70,000 within BC, LP, LC, and SCs. And this just gives you an example. Here's the heterozygote variant, and here's the gain in this particular case of H3K4 trimethylation with the luminal cell associated with this variant. So we have this collection of variants, and we asked, is there a way we can validate this? So again, this is a matrix of the women, of the individuals that were used in this particular case. And this individual is the individual that contains this heterozygote position. And we can see that there are cell lines, MCF10A, for example, that doesn't have the reference, and we have cell line, or that doesn't have the heterozygote position, and these H-TERT cell lines that do. And when we looked at the expression, indeed, we could see that even within the cell lines, and those in the light gray here, that we saw an agreement when the heterozygote position was present, we saw an increase in expression of the nearby gene. And in this particular case, the gene that this heterozygote position exists within the promoter is ANXA1, and it's been associated with breast cancer survival, in particular subtypes of breast cancer. So for example, in this breast intrinsic subtype, you can see that's actually has a very high hazard ratio. So we then went into the MCF10A, which was homozygote for the reference position, and using a CRISPR-Cas9 strategy, went in and did a precise edit of that variant. And this shows you that in the original pool that we obtained, the frequency was very low. We then sorted for 100 cells across a well to increase the frequency of the heterozygote position, and then from there did single cell cloning to generate a series of cell lines that contained only that heterozygote position. And this is just my last data slide to show that indeed we were able to recapitulate the relationship between this heterozygote allele and expression of ANXA1, suggesting that we may have identified a cell type specific or cell type specific interpreted heterozygote variant within the luminal cell population. So just to summarize, so I hope I've convinced you that the EpiATLAS provides a reference for human epigenomic research, that we generate standardized experimental and analytical workflows, and these are designed to generate and share reference epigenomic maps for primary human cell types, that biological replicates provided for a probabilistic framework, as I showed from Jonathan's work, and then from Axel's work, that genetic variants in association with cell type-specific epigenomic states are common in human breast cell types. And just to say, in terms of a call to action, and EpiATLAS as we look into the future, that really mature experimental and computational tools are being developed and are developed for epigenomic reference mapping. We have an ecosystem for data submission and sharing available. However, there are many tissues and cell types, and this just shows, this vignette here in the middle just shows some cell types that have not been sampled at all within the IHEC reference. So really what we need to do as a community, arguably, is additional programmatic efforts and even efforts within individual research groups to continue to refine and extend the EpiATLAS. We need to finish what we started and to finish defining the reference maps for all human primary cell types that are amenable to these assays. And really, I think about this in the same way that we think about the human genome. It took us 20 years to complete the human genome, arguably to the T2T assembly. And I suspect that in another 10 years, or I hope in another 10 years, that we can look back and say that we have generated a reference epigenome maps to enable the research community. Okay, and just to say it takes a team. These are the individuals that were involved in my group in terms of the processing of the data that I showed you today. And it really does take a team to do this kind of large scale reanalysis. And just to thank the funders and my dear collaborator, Connie Eves, who I worked with for many years on the breast model, and indeed the cell types that I showed to you today. So with that, I'll stop and be happy to take any questions.